Hello students, today we are going to study about chapter 6 molecular basis of inheritance. Myself Madhulika Jain. So, let us have a look on few important definitions. Genome, sum total of gene in haploid set of the chromosome, that is called as a genome. Codon, a sequence of 3 nitrogenous bases which is present on DNA and that codes for one amino acid, that is known as the codon. Anticodon, a sequence of 3 nitrogenous bases on transfer RNA which is complementary to the codon on messenger RNA, that is called as anticodon. Satellite DNA, the repetitive sequence, DNA sequence which makes a larger portion of genome and have high degree of polymorphism, but it does not code for any protein, that is called as the satellite DNA. Now, there are some regions on messenger RNA, that is exon and introns. Exons are the genes which become part of messenger RNA and they code for the specific protein. Introns, the region of gene which is not used for the formation of a protein and this is removed during the process of messenger RNA or splicing. Now, what is splicing? Splicing is the process where introns are removed and exons are joined together to convert heterogeneous nuclear RNA into messenger RNA, which is required to make one specific protein. DNA polymorphism. It is the variation at the genetic level in the base pairs where an inheritable mutation is observed in a population on a very high frequency. Students, now look at some full forms, that is VNTR, variable number of tandem repeats. Then YAC, that is the yeast artificial chromosome, BAC, bacterial artificial chromosome, SNPs, that is single nucleotide polymorphism, HGP denotes for human genome project, and heterogeneous RNA, that is heterogeneous nuclear RNA, hnRNA. Students, now look at how the DNA or the deoxyribonucleic acid is formed. We know that DNA is a polynucleotide chain and a chain has, this chain is made up by repeated sequence of nucleotide and one nucleotide is made up by 3 components. First is the nitrogenous base. Students, we know that there are 4 types of bases, that is adenine, guanine, cytosine and thymine. Adenine and guanine, they belong to purines and cytosine and thymine, they are the pyrimidines. Another group is pentose sugar, that is deoxyribose sugar in DNA, and the third one is the phosphate group. The DNA model is that was proposed by Watson and Crick and it is commonly called as Watson and Crick model of DNA. They proposed the double helical structure and they also said that DNA is made up of 2 polynucleotide chain which are coiled to each other and their backbone is made up of sugar and phosphate and the nitrogenous bases are projected towards the inside. 2 strands in DNA, they are arranged in the manner that they are anti-parallel. It means the one chain has polarity to 5 prime to 3 prime and the other chain has the polarity from 3 prime to 5 prime. These 2 strands of chain, they are held together by hydrogen bonds, and A and T. Adenine is always bonding with T, that is thiamine, with double bond, and cytosine always bonds with guanine, that is with triple bond. The pitch of the helix, that is one turn, that is 3.4 nanometer, which contains 10 base pairs in each turn. So here students, you can have a look on the structure of single strand of DNA that shows how these nucleotide units are connected to each other. Now DNA is so long and it is packed very compactly. So how it is packed? We know that average distance between two base pairs, that is 0.34 nanometer or 34 angstrom. So if we know the total number of base pairs in DNA, we can calculate the total length of the DNA by multiplying total number of base pairs with distance between two consecutive base pairs. In prokaryotes, DNA is compactly, firmly present into the center only. This is not scattered into the whole cell. And that is being negatively charged. It is held by some positively charged protein, and that is why it is retained into the center. In prokaryotic cells, just like in bacteria, the region of cell which is having the DNA, that is called as nucleoid, and in the nucleoid region DNA is present in large loops. Let us look at the packaging of DNA in eukaryotic cell. There is a set of positively charged basic proteins. These proteins are called as histone proteins. Each histone protein that contains 8 molecules, and they are combined together and now known as the histone octamer. The DNA which is negatively charged, that is wrapped around the histone octamer, and that structure is called as the nucleosome. Outside that nucleosome is having the wrapped DNA. Outside of that, there is some DNA in the linker region. So there are repeating units of nucleosome. These nucleosomes, they constitute repeating unit of a structure in nucleus, and when these repetitive units are present, that structure is called as a chromatin. Students, you must have seen that beads on the string structure in chromatin, that is packed to form chromatin fiber, that are further coiled and condensed, and that happens during metaphase stage of cell division. There are some additional sets of the protein referred as non-histone proteins or NHC. They are required for further supercoiling of the chromatin material. So when chromatin material is packed and it becomes very compactly packed and it gives dark stain, that chromatin is called as the heterochromatin. This heterochromatin part does not transcript, does not code for any protein, but there are certain regions that are loosely packed and they give light stain, that is called as the euchromatin, and that is transcriptionally active part of chromatin. Euchromatin is transcriptionally active part. Here we can have a look on how is the nucleosome formed. Here you can see in the center that octamer is present, and towards that, over that, DNA is wrapped. This is one nucleosome, and these repeating unit of nucleosome, they make the chromatin fiber. Thank you.